Welcome to the video recording and audio recording of the Chem 344 Molecular Modeling Prelab lecture. This is the first semester that we're using our new cluster Phoenix, which is pictured here showing the hardware, to do calculations using WebMO to support the research that we're doing in the Chem 344 experiments. This first experiment, Experiment 5, is designed to get you familiar with the types of calculations you'll need to do throughout the semester, as well as the type of reasoning that you'll need to do to interpret these output files and these images that you're going to see and make some chemical sense of them. Here we have a biphenyl molecule with a nice dihedral twist between the aromatic rings. You're going to see that in a future experiment. This is caranuline, and we're going to talk about that in a little bit today. This is anisole, and a predicted IR spectrum of it. And these are the types of images and information you're going to get used to seeing today and throughout the 344 semester. First, a little background on what computational chemistry is actually used by organic chemists. Um, Typically, those of us who are involved in this type of research predict a lot of geometries, and everything else that we do is kind of dependent on how well we can predict the geometry, because the predicted geometry gives us information about physical properties, the volume of molecules, the orientation of the atoms in the molecule, the density, the dipole moment, the rotational constants, all of these important physical properties. We're always concerned about the relative energies of different structures. We do a really nice job of visualizing the electronic structures of molecules using computational chemistry. By that I mean taking a look at the molecular and atomic orbitals, and we're going to talk about how we do that throughout the day today, and you'll be doing that during the semester. Computational chemists do a lot of work predicting the regio and stereochemical outcomes of reactions. You're going to do that in a couple of the experiments in Chem 344. We're going to spend some time today and throughout the semester looking at the stabilizing and destabilizing interactions of parts of molecules. Computational chemistry is great at helping visualize those. Computational chemists predict all sorts of observable experimental spectra from computational results and the optimizations of these predicted geometries. In Chem 344, we'll concentrate on the IR or NMR spectra, but organic chemists also predict UV-Vis as well as microwave or millimeter spectra. My work, we do a lot of predicting of microwave spectra. We're also going to use some computational chemistry to take a look at the transition states of chemical reactions and actually look at some chemical pathways for how reactions can proceed. One of the real benefits of computational chemistry, aside from the support of experiment, which is sort of these regiochemical outcomes, the spectroscopic properties, physical properties of molecules, is our ability to gain insight into transition states, which would be not possible without this sort of modeling. Transition states are species that occur partway through a chemical reaction that last for a time scale of one molecular vibration. Really, we have no good way to spectroscopically to probe those transition states, but we can try to make sense out of them using some computational chemistry. And you're going to look at that later in today's experiment when you're looking at the SN2 reaction, which is the final computational exercise. So before we get into today's stuff and working through how to do the calculations, I wanted to walk through several examples of how chemical properties and aspects of molecules can be probed using computational chemistry, and just give you some insight into how we need to think about these things. And so I started out with some really simple examples. I wanted to look at hydronium ion and acetate ion and see how your own background from general chemistry impacts how you think about these molecules and how we can gain some additional insight doing some molecular modeling. So I have the Lewis structure for a hydronium ion drawn here. I have a formal positive charge on the oxygen. And I think this is a great example of how oftentimes people learning general chemistry and then into organic chemistry have really inappropriately internalized formal charge. Right when you learn formal charge, you are told that it's a formal bookkeeping mechanism for keeping track of electrons in Lewis structures and not necessarily indicative of any real charge in the molecule. Computational chemistry can give us some insight into exactly where the charges lie in a species like this. First, I'm showing you here an optimized geometry. The computers determine that that's the structure of hydronium ion. It actually can predict where the charges are in that molecule. And as you can see, despite the positive formal charge in the oxygen, it actually has about a minus 0.82 charge on that oxygen atom, with the rest of the positive charge being distributed amongst the three hydrogens. This results in a polar structure with a dipole moment of 1.7 dBi, and the dipole is along this axis where we have a negative end up here by the oxygen, a positive end down there by those hydrogens, and you can actually ask computational programs to map the electron density for you, and they do a red depiction of the negative part and blue for the positive and a color gradient in between. And you can see that, yeah, this is a polar molecule with a red negative end and a blue positive end. We can do the same thing for acetate ion, where we have two possible resonance forms, and you should remember really well from Gen Chem that we have a negative formal charge on this oxygen and a negative formal charge on this oxygen, and really there's a negative half a formal charge spread between the two, because the true structure of acetate is a combination of these resonance forms. So we'll do a quick calculation on acetate. I optimized the geometry using WebMO and asked it to show me the charges, which I have listed on each species. And I'm using the WebMO convention of red for negative and blue for positive here. And you can see that, not surprisingly, the formal charge of -1, sort of split between the two oxygens, giving about -1/2 at each oxygen, isn't quite right. And you should have been able to predict this from your own knowledge of Gen Chem. You know that oxygen is a highly electronegative atom, and in that way will pull electrons towards itself. It does so in the hydronium species, taking the positive formal charge and making it a considerably negative charge of 0.82. And it does so here in the acetate species, pulling extra electron density towards itself to bring its formal charge from what might be about -1/2 up to something more reasonable like -0.8. A kind of interesting insight here is the two carbons in the molecule have actually opposite charges. The carbon that's part of the carboxyl group is positive, and that you'd expect by those electron withdrawing oxygens attached to it. The carbon alpha to that actually has a negative charge. This sort of negative charge character is actually an important thing in terms of chemical reactivity of these types of species. And of course, the hydrogens get a positive charge, which we're going to see as a common theme in organic species. This results in a pretty hefty dipole moment, pulling electrons in this direction towards the oxygens. And we can see that in a nice electrostatic potential map that WebMO is able to draw for us. So we can gain additional insight into both structures by getting some optimized geometries, as well as where the charge distribution is in the molecule using computational chemistry that may not always be obvious from our Lewis structures and our formal charges. And one thing I want you to remember throughout the semester for Chem 344 and throughout your chemistry career is that formal charges are not real. They're just bookkeeping mechanisms, and you need to be highly suspect of them and really, really go out of your way not to internalize that oxygen is positive or that these oxygens have a half a negative charge, because neither is really physically meaningful. Another little insight we can get looking at charge distribution is for some aromatic species, and these are sort of important to my work, and I'll explain a little bit more about that as we go through this. This molecule is benzene. You're well familiar with it. You should be familiar with this sort of an image from your textbooks that show a blue or positive outer part where the hydrogens are located, and a red negative inner part where the electron density of that aromatic system is adding to some electron density, as well as our slightly more electronegative carbon atoms relative to our less electronegative hydrogen atoms, result in some good electron density in the center shown in red. Since this is a really nice D6H symmetry, where all of the atoms are opposed by an identical atom on the opposite side of the ring, results in no dipole moment, which is sort of sad for folks in my type of research. We're really interested in what kind of organic species might be found in space And radio astronomers use big dish telescopes like this one to make those searches. In order for a molecule to be detectable by radio astronomy, it needs a dipole moment. So that takes sort of benzene out of the mix of possible molecules that we could detect using this technique in space. Um, this picture over here I took myself when I was at the Green Bank Telescope, which is named after Robert C. Byrd, a late senator from West Virginia. I think most of West Virginia is named after him at this point. And if you look at the bottom of this photo, you can see a group of astrochemists getting a tour of the telescope. The dish itself is so large that you could put a football field on it and have room to spare. And so this is a giant, giant telescope, and it's used to search for molecules in space. Just because we can't find benzene doesn't mean we can't find other organic aromatic species. One of the ones that's been searched for is caranuline. And using the same type of electrostatic potential maps I did for benzene, I can show you what that structure looks like. Notice there's not the deep red that we see in benzene, but there definitely is a negative inner part of the molecule and a positive outer part of the molecule. The exciting bit about this is when we optimize the structure of caranuline, we see that it is a bowl-type species where it has a raised center, and because that center is negative and the lower outer part is positive, it actually creates a pretty sizable dipole moment for an aromatic organic species like this of 1.7 dBi. So this molecule is a prime candidate for detection by radio astronomy, which would be really exciting because right now there are only a couple of very small aromatic molecules like cyclopropanone that are known in the interstellar medium, and there's only about 170 molecules in total, and it'd be great to start adding some organic aromatic species to that list.